Thank you everyone who is joining us. We have a few announcements as everyone joins. First, we are recording this session and it will be available in Whova, our conference app, within 2 weeks after the Congress ends. If you'd like to ask our speakers a question during this session, please use the Q&A area to the right of your screen. The chat window is where you can engage with other attendees. And we kindly ask that you keep your microphones and cameras off during the presentations and follow the presenter's lead on when to engage.
I'll be tuning into the Congress now.
Okay.
It looks.
Thank you for letting me know. I'm sorry I've been bugging you all.
It's all right.
I think your mic's on, sir. We kindly ask that you keep your microphones and cameras off during the presentations and follow the presenter's lead on when to engage with them. It looks like we're ready to get started, so I'm going to hand it over to Kevin to get us going.
Thanks so much, Melly, and welcome everyone to the biodiversity session. Our first talk is going to be by Jamie Woolet. She's a master's student at Colorado State University in the Warner College of Natural Resources. And she's going to be talking to us about effects of 3 wildfires on habitat use by birds that are critical for maintaining pinyon-juniper woodlands through seed dispersal. So welcome, Jamie.
Thanks, Kevin. I'll share my screen now. All right, hi everyone. Like Kevin said, I'm Jamie Woolet, and I'm a master's student at Colorado State University. And I'll be talking about the effects and implications of fire on pinyon-juniper woodland bird communities and tree regeneration. So changing fire regimes and forest resilience are a growing concern in the conservation and persistence of our forested lands. And in pinyon-juniper woodlands, which are a widespread forest type in the southwestern US, fires typically have been classified as infrequent and stand replacing. However, ecologists have identified shorter fire rotations occurring in many stands compared to pre-Euro-American settlement. And changes in fire frequency are of growing concern due to drought, invasive grasses, and human-caused ignitions. This is concerning because these pinyon-juniper woodlands require these very long fire-free intervals in order to fully recover into a woodland again. However, additional stress due to drought, like what we've been seeing the past couple of decades, combined with more frequent fire, leads to uncertainty regarding the regeneration of these forests. And it's important that these forests do recover because as a dryland forest, they provide key ecosystem services in the southwestern US and have cultural, economic, and ecological significance. Regarding their ecological significance, this system has a very important relationship with the birds that live there. So these woodlands provide breeding habitat for over 70 species of birds across its range, about 18 of these being obligate or semi-obligate, meaning that they only breed in that landscape or one other. And on the other side of this, following wildfire, woodlands regenerate with the assistance of seed dispersing birds that retrieve seeds from living trees and cache them elsewhere, up to thousands of kilometers away. So being able to reach the center or near the center of these large burn patches. So knowing this ecological relationship, I noticed an issue within the current literature in that there's few studies that describe how fire affects these bird communities. And similarly, there's little information about what drives pinyon and juniper seedling regeneration success or failure in these fire-affected areas. So my master's work hoped to address these 3 objectives. 1, how does pinyon and juniper seedling regeneration differ in burned, unburned, and refugia? And how is this impacted by forest structure? 2, how does bird community structure compare in burned, unburned, and refugia? And lastly, does the presence of seed dispersing birds affect seedling regeneration in the burned areas? So to tackle these questions, I selected 3 fires that occurred in Western Colorado. One that burned last year, the Pine Gulch Fire, which for a brief stint was Colorado's largest fire, but was quickly overtaken. And then 2 others that had burned in the mid-1990s. And I'll refer to these as a recent burn and the 2 old burns. Within each fire perimeter, I looked at fire severity maps and selected burned patches, refugia patches, so unburned islands within the fire perimeter, and then unburned areas outside of the fire perimeter as a reference. And these patches were later ground truthed in the field to ensure that, one, they were large enough to successfully put the point count stations, and then also that fire had occurred in these patches. So in total, there were 6 treatment types: old burned, old refugia, old unburned, and recent burned, recent refugia, and recent unburned. And so across all of these treatment types, there are 32 forest inventory plots and 126 avian point count stations. The forest inventory plots were 0.02 hectare circular plots where we measured slope, aspect, and elevation at plot center. We measured canopy cover, understory cover, substrate cover, and surface fuels along 4 transects in the plot. And then we measured tree characteristics. So we counted, identified the species, and measured the diameter root crown of all of the trees within the plot. And then we also counted all of the seedlings and determined size classes for all of the seedlings within the plot. For the avian point count stations, we did 6-minute point counts, starting at the break of dawn and going until about 10:30 in the morning. And we recorded all birds seen and heard, as well as their detection distances. And these stations were revisited 3 times throughout April to June. So jumping into the results for objective 1, how does pinyon and juniper seedling regeneration differ in burned, unburned, and refugia? We did not detect, or we did not note any pinyon regeneration at any of the burned plots. And only 1 old burned plot had juniper seedlings. So generally the burned areas had very low regeneration spotted. And so then instead I compared the refugia plots and unburned and noticed and detected no difference in seedling regeneration, just juniper regeneration and just pinyon regeneration across any of the treatment types. For the second part of this objective, how is regeneration impacted by forest structure? I tested the effect of stand characteristics on seedling regeneration in refugia and unburned by running generalized mixed models with all of the non-correlated variables that were measured, and selected the best fitting model for each of these seedling classes. So all seedlings, pinyon seedlings, and juniper seedlings. And a few themes arose. So elevation had a positive relationship with each seedling category. So as elevation increased, regeneration also increased. Shrub cover had a negative relationship with each seedling category. So as shrub cover increased, the number of seedlings recorded decreased. The total number of live trees had a positive relationship with all seedlings and pinyon seedlings, and the number of live juniper trees was positively associated with juniper seedlings. And lastly, tree basal area had a negative relationship with all seedlings and pinyon seedlings, and live juniper basal area had a negative relationship with the number of juniper seedlings. And so moving on to the bird results, I'll preface these next couple slides by saying that this is preliminary results. So generally when quantifying avian abundance, you have to detect, or you have to account for detection probability. And so I'm still working on this. And so what I'll be presenting on is just my detections from the field, not accounting for detection probability. So with that being said, species richness was not significantly different across treatment types. And species diversity, the Shannon diversity, is not different across treatment types either. However, for the diversity, if you look at just all the old treatments versus the recent treatments, recent had a higher diversity. So then I decided to look more at what was going on within the specific guilds. And so I divided guilds up by obligate birds, semi-obligate birds, pinyon seed dispersing birds, and juniper seed dispersing birds. And again, I didn't calculate statistics here because these are just my field observations. However, I think it shows an interesting trend, or at least an interesting story about what could be going on within this system. And so when looking within fire age groups, so old fires versus recent fire, we see pinyon-juniper obligates and semi-obligates less in the burned areas. And again, comparing the old fire and recent fire for pinyon and juniper seed dispersing birds, we see a much higher detection of pinyon seed dispersing birds in the old burn and a much higher juniper seed dispersing birds in the recent burn. And so most of the pinyon dispersing birds were, I think it was like 98% were woodhouses scrub jay. And then for the recent burn, these are mostly American robins and mountain bluebirds that were the juniper seed dispersing birds. So for this next objective, I have a question mark for now. And this objective was originally in place to tie together seedling regeneration in burned areas and seed dispersal, with the thought that if there was regeneration in a burned patch away from a live seed source, it had to have been cached there by either a bird, or if it was closer to the edge, then possibly a rodent. And when I designed this study, I did anticipate there being some seedling regeneration in the older burn sites. However, this was not observed, even though there was a larger number of detections of woodhouses scrub jay in these old burned areas, and a much higher detection of juniper seed dispersing birds in the recent burn. However, it is comforting to see these observations because it may have important implications for future regeneration. So to conclude, there was very little seedling regeneration observed in burn sites at any age. And anecdotally, while walking in between sites, occasionally we would see a juniper seedling in the burned areas, but we never noted seeing pinyon. This isn't surprising since juniper are known to establish before pinyon do. However, looking into other burned pinyon-juniper woodlands and further monitoring these specific sites would be interesting to determine when, under what conditions, and where we begin to see more regeneration occurring. And in neighboring unburned areas, so unburned as in the unburned reference sites outside of the fire perimeter and the fire refugia, seedling regeneration success was linked to increases in elevation and more live trees, whereas less regeneration was linked to higher shrub cover and more tree basal area. It is good to recognize that in general, there are seedlings establishing within the same geographic area as the neighboring burned areas. So it may be possible that birds are not actively moving seeds into these burned regions. Or it's also possible that there's a lack of optimal microhabitats conducive to seedling regeneration or seedling germination and later survival. It's important to note that this region has been under a severe drought for the past couple of decades, and across sites, the difference in elevation ranged about 524 meters. So it's possible that higher elevations may be offering a buffer to climatic stressors for these seedlings. Bird richness did not change across treatment types. However, the community composition when looking at the guilds do shift. And lastly, bird diversity is higher in the recently burned or in the recent treatment area, which may be a proponent of biodiversity, creating these very different landscapes and causing a more variable bird composition. My next steps will be to properly assess the bird abundances and look deeper into these shifts in the community with respect to habitat type, in order to hopefully paint a more descriptive picture of how fire affects these bird communities. And so with that, I'd like to thank my advisor and my committee for guidance and help with the study design and development, my awesome field crew for all of their hard work this past summer, the Joint Fire Science Program for partial funding for this work, and the Grand Junction BLM office for being a really helpful point of contact throughout my field season. My contact information is at the bottom, and I'd love to hear from you for any questions or suggestions. Thanks.
Thanks very much, Jamie. Nice job. We don't have any questions in the queue yet. I'll keep an eye on that. In the meantime, I had a question. Do you feel like there are any kind of positive feedbacks about the burn that are actually attracting the birds to occupy those spaces behaviorally? Because it's kind of surprising that there's almost no vegetation and yet the birds are showing up there at about the same rate. Is there something to compensate the fact that there's seems to be very little for them there?
Yeah, so with the recent burn, it could just be like the very big openings. I need to look more at the life history of these specific responding birds and see like if they enjoy these more open habitat types, but that could be it. And then for the older burns, there was quite a bit of shrub cover around, so it could be these like more shrubby enjoying birds that are there. I think the Woodhouse's scrub jay does like these dynamic structures, so that could be why they're hanging out there more.
Okay, we have a question from Wyatt on the panel. Was there more an invasive species of birds in the older burn areas, which may have attribute to the lower diversity?
No, so we did not see any like invasive birds. So it was mostly like the birds that I had expected to see that were there. But yeah, no invasive birds.
We have a question from Sean McKinney. A nice talk, wondering if you're observed any seed caching in the recently burned areas.
Not that I could tell. We didn't really see very many like scatter hoarding birds in the recent burned area. And I wasn't really looking like at the ground for any specific seed caching, no.
You're just observing the presence of the birds pretty much.
Yeah.
Yeah, it's interesting. I have a graduate student who is looking at the effects of fire attracting birds and therefore seed dispersal in the longleaf pine ecosystem, but it's a lot easier to understand the mechanism there, where most of the plants are perennial and are resprouting, and a lot of them flowering in response to the fire. So there's an increase of resources there, but that didn't necessarily seem to be the case in the pinyon-juniper lands. Are these fires? I mean, this may be a obvious question, but are they more severe than they were historically? Is there a big difference, do you think, in the pattern of fire?
So severity, no. So usually, if a fire were to catch and persist in pinyon-juniper woodlands, they would be totally like stand replacing. And so severity is not really like the issue researchers are seeing with PJ woodlands. It's more the frequency of occurrence. That's more the concerning factor, and possibly the size with how large the Pine Gulch fire was comparatively.
Which is important because of the spatial, the distance for dispersing the seed. Yeah,
exactly.
I think we have time for one last question from Thea Whitman on the panel. Say, great talk. For the obligate birds, do they also disperse other non-juniper or non-pinyon seeds? And what are they likely eating in the plots with very little regeneration?
Yeah, so for the non-obligate birds, there's a lot of grasses and shrubs that are also there. And so they're probably consuming those, and like the the juniper is like a really desirable berry. So a lot of birds tend to, at least frugivorous birds, enjoy like eating their seeds.
Mhm. Okay, well, thanks again, Jamie. I think it's time for us to move on to our next talk.
Thank you.
So we'll bring on Wyatt Bagwell next.
All right.
And Wyatt's a graduate assistant at the Stephen F. Austin State University, Arthur Temple College of Forestry and Agriculture. And he's going to talk to us about effects of prescribed fire and nutrient availability in deer browse within the historic range of the longleaf pine in eastern Texas. Welcome, Wyatt Bagwell.
All right. Can you all see that?
Yes.
All right. Well, good morning to everybody, and thank you for the introduction. And thank you all for attending this talk. First, I'm going to give you all a little bit of background about white-tailed deer in East Texas. Uh-oh. Okay, there it goes. White-tailed deer are an important game species in East Texas, often producing millions of dollars in revenue every year. And to manage white-tailed deer in East Texas, there's several different options. However, prescribed fire is often the most easiest and most common to use. And other studies have noted that's been used to improve browse availability, forage production, and nutrient availability for white-tailed deer. So a basic white-tailed deer diet is that they eat the leaves, flowers, fruits, and twig ends of hardwoods and shrub species, and fleshy fungi, and the stems and leaves of forbs and grasses. And also the utilization of those very seasonally. And that's primarily because what it's eating also occurs seasonally as well. And a diverse diet is required to obtain those essential nutrients that it needs. Also, I do want to point out in this picture, as you can see here, that's just eating the stem ends here. And that's because that's where the most nutrients of the plants are. So the majority of a white-tailed deer's diet is browse, and it's usually divided into 3 different choices or classes, with first choice being the most preferred and nutritious. And a good example of that is green briar. Has high protein content, is very easy to digest. Second choice is a lot more abundant and not as nutritious, and it's usually harder to digest. A good example of that is yopon. It's pretty much everywhere in East Texas. In fact, it was in all my plots, just about. And third choice is the lowest quality and least preferred. Usually these have around 0% utilization, and if high utilization of this is noted, then other management actions need to be taken to either reduce the deer herd or increase the habitat or improve the habitat. So nutritional requirements for white-tailed deer vary with age, physiological state, and environmental conditions. For example, a mature lactating female with a fawn requires about 14% crude protein in its forage. And also in the South, forage availability is very abundant down here. However, the quality of it is often lacking. And then deer, they also require a diverse diet to meet those needs, and those diets vary in composition and concentrations per species, and how those species are and also placed in the environment can affect how a deer spatially utilizes it too, in order to satiate its needs. Even though browse might comprise the majority of the white-tailed deer's diet, forbs are the most preferred. However, they do even have preferences on forbs themselves. For example, most legumes are usually highly preferred by white-tailed deer due to their high protein content. A good example of one that is not preferred is goldenrod or solidago species, just because it's very hard to digest. So the objectives of the study was to assess the impacts of different fire frequencies and seasonalities on the utilization and nutritional value of preferred white-tailed deer browse species, and examine the relationship between browse utilization by white-tailed deer populations and time since prescribed burn and season of burn, and determine which prescribed burn frequency produces the most preferred herbaceous biomass for white-tailed deer in East Texas. So for study sites, I have 5 different study sites. However, today I'm only going to be reporting a portion of my results. And I'm going to be talking about the Winston Land and Cattle Ranch and the Nature Conservancy's Roy E. Larson Sandyland Sanctuary. All of these study sites occur in East Texas in the Piney-Woods ecoregion. So a little bit of background about the Winston Ranch. They primarily manage for deer, cattle, and timber. It has 8 plots. There's 8 plots that I did in there, and it was located in Nacogdoches County. The dominant overstory is longleaf pine, and it's also a relatively young stand as well, about 16 years old. And because of that, it also had a mean canopy density of 73%. Another factor I'd like to point out is that the stands were planted in rows, and that too can affect how browse species pop up. And also the predominant soil texture is fine sandy loam to clay loam. And the reason why I'm mentioning that is because that can influence cation exchange capacity in the soil, meaning that's the amount of nutrients that the plant can pull up from the soil, which goes up into the foliage, and the deer eat the foliage. And it had an annual mean fire return interval. Sandylands was the other study site that I did, and it was located in Hardin County. And it's a non-government organization, so its primary goal was just to conserve natural areas. And 14 plots were utilized here, and the mean canopy density was 28%, and the dominant overstory was longleaf pine as well. As you can see from the picture, it's much older stands, and hence the name Sandyland. The soil texture varied from sand to loamy very fine sand, and the annual mean fire return interval was 1 year. Also in 2020, because of COVID, no burns were conducted. So I was able to survey 1 year since burn on the vegetation, and also 2 years since burn on the vegetation. So in order to achieve this study, the plots selected for Winston were randomly selected, and they're 100 feet off the road, and also make sure they were not near any deer feeders or protein feeders. And the plots for Sandylands were based off of pre-existing monitoring points, and they're also made sure to be 100 feet off the road or more than that. And browse utilization was assessed using a modified version of Texas Parks and Wildlife Department stem count index method, and this occurred in the winter prior to the growing season. And also, while the browse surveys were being conducted, nutrient samples were also being clipped, and what was being clipped was essentially to mimic what a deer would eat, grabbing all foliage, green foliage, and also stems, buds, what the deer, essentially trying to mimic best we could what they're eating. And also these samples were sent to Dairy One Forage Laboratory in Ithaca, New York, following their lab forage sampling procedure. The other part of my study, which occurred in the growing season, was sample herbaceous biomass. And these occurred on the previous sampled plots for browse in the winter. And the sampling frame that I used was a 0.3 meter by 0.6 meters by 1.5 meters. And the bottom portion of the frame was essentially a Dobmeyer frame. And in their cover classes of each individual species was determined. And once that was done, all species were clipped and then placed in separate bags. And then they labeled by species and by plot, and they were then put in a drying oven and weighed to the nearest gram, weighed to the nearest 100th gram. Also, the vertical portion of this that goes up to 1.5 meters, that's to mimic how high a deer can reach. And that's used, for example, if there's a shrub in there, such as yopon bush, all foliage would be removed off that, and new growth, and that too would be placed in a bag and labeled by species, and then placed in a drying oven till dry weight remained constant. After all their weights were dried, those were multiplied out to kilograms per hectare. So for data collection, what I have done is I conducted browse surveys in the winter of 2020 and 2021, and also collected browse samples during that time. And the herbaceous biomass was sampled in the summer of 2020 and 2021. So for my data analysis, I performed kernel density plots, PCAs, Shapiro-Wilkes tests, and RStudio version 4.03 on browse utilization and nutrient availability. And I performed the in-pairway procedure in SAS version 9.4. Test significance of utilization by site, by year since burn, and preferred herbaceous forages were also tested for significance with the same independent variables. I do want to mention why I use the in-pairway procedure in SAS is because all variables failed the Shapiro-Wilkes test for normalcy. And then here's my results for browsing nutrients. So utilization, the kernel density plot, as you can see, a lot of the utilization of browse species occurred between 0 and 10%. This x-axis right here, even though it says 0.1, that's actually 10% utilization. And then since utilization does vary by choices, I subset the data and made kernel density plots for all those. I can see over here, the majority of the data for this green line right here is third choice, and it occurred between 0 and 10%. And then first choice had a utilization which is the red line, which occurred all the way up to almost 60%. And then second choice, which is the blue line, and that occurred in between first and third choice. Then I also performed a principal component analysis, or PCA, to see the variability of the data. And I do realize it's hard to read, but the next slide should clear those up. With the combined data set of Winston-8 and Sandilands, a lot of grouping between the second and third. The blue is the third, and green is second. And then Winston-8, there's a lot of grouping over here by the adjusted crude protein, which is the green. And then for Sandilands, there's also a lot of grouping between adjusted crude protein, second and third choice over here. And also these variables, which is this large clump that's hard to read, is all the net energy calculations for nutrients. So to make it easy to read and understand, I looked at the contributed weights of each of the variables in R. So the contribution of variables for dimension 1 or axis 1, as you can see here, the 5 most important or held the most weight were all had to deal with energy. And then the contributions for variables for a second dimension all had to do with essential nutrient, macronutrients, or micronutrients. Now this, the variables of the second dimension did variable when it was subset by site. Winston-8, relative feed value actually was the most important variable and leading for that variation. Whereas for Sandiland, phosphorus was actually the most important variable in the second dimension for leading that variation. Then I performed in-pairway results for utilization and SAS. And the only thing that was significant for utilization by site, it was significantly different between the two sites. And also utilization by year since burn was significant. All the other blanks right here, it was not significant by adjusted crude protein by site and total digestible nutrients by site, nor the net energy for growth and maintenance by site. And then Sandilands, utilization by year since burn, that was not significant, nor any other tested variables. And the reason why I chose to test these few variables, adjusted crude protein, total digestible nutrients, and net energy for growth and maintenance, is because that's probably what most land managers would be concerned about. And then the results for the herbaceous forage, I performed similar same tests in pairway and SAS. And the preference by site, the percentage coverage of her preferred herbaceous forage, that did significantly differ. And Winston-8 did have a higher percent coverage of preferred herbaceous forage. Whereas and this also held true, and year since burn, it was significantly higher. And the preference by year since burn, it had a higher mean too. And that does coincide with other literature saying that 1 year since burn, the preferred herbaceous forage should be higher. Also, I would like to give a special thanks, or I'm currently thanking all those who helped. I'd like to thank the McIntyre Stench Grant for funding this project, my advisor, Dr. Oswald, my committee members, Dr. Glasscock and Dr. Kidd, and all fellow graduate and undergraduate students who helped with the field work and some of the lab work. And I'd like to thank Texas Parks and Wildlife Department, the United States Forest Service, and the Nature Conservancy and the Winston 8 Ranch for allowing me to conduct their studies on their properties and for the data that I've obtained from them. Any questions?
All right, thanks a lot, Wyatt. Great presentation.
Thank you.
I don't have any questions in the chat lineup yet. I had a question. What do you think the mechanism is for the higher nutrient content in the 1 year post burn versus 2 year in the herbaceous vegetation, given that the herbs die back every year anyway and kind of start from fresh in the spring, right?
Yes. So in my study, in my research, I've actually found out that there's some studies that suggest that since the browse or the herbaceous or the shrubs are perennial and they have much deeper roots, that the fire is actually not affecting the nutrients that much. However, due to it burning back and fire stimulating new growth on the plants and deer eat the new growth, that's where it gets the majority of the nutrients. And then for the herbaceous forage, that's pretty well documented that after 1 year since burn, it does have higher nutrient. Did I answer your question?
I think so. So it has to do with the fact that they're eating more recently resprouted vegetation,
Yes.
as opposed to just vegetation that's greened up in the previous spring or beginning of the growing season.
Correct.
So it's basically utilizing those carbohydrates that have been brought up from the underground storage in the plants. Interesting. Did you notice any difference in species with the woody resprouting plants in terms of was there a higher nutrient level in the ones that were resprouting more vigorously or larger? There's generally a thought that the the native upland broadleaf woody plants that are historically part of this ecosystem tend to be kind of slow resprouters that keep a lot of energy in the roots and have a high protein content and don't have as high of a, not have as high of a photosynthetic rate. But do you think like the deer browse is higher on things like sweet gum or water oak that are?
Yes, there's actually an
resprouting
instance.
faster?
I wish I could have performed a nutrient analysis on it, but there's a water oak, which is generally third choice of preference. And there's 130 stems that were counted on it. I made my technician count it anyways, just because they're interested. And over half of those stems were utilized or browsed by deer. And water oak, like I said, is usually third choice. So that was just something phenomenal to me. And all those stems were new growth.
That's interesting. So the mesification process might be increasing browse for deer, but maybe the deer providing some kind of feedback mechanism against those species.
Yeah.
One could only hope, I guess.
Yeah.
Because I don't think the burn managers like sweet gum and water oak that much
No, they
in
don't.
terms of the flammability and ease of management from a fire perspective, right?
Also, I do want to mention one other thing. Yaupon might actually kind of be a necessary evil because majority of my plots, they all had yaupon in it.
Mhm.
And if you've ever been in Southeast, it's pretty much everywhere. However, since that is second choice, that's what's available for the deer to eat. So you might be creating more nutritious yaupon for the deer to eat.
Yeah, and yaupon is also very flammable, so it blows up when you burn it, which is kind of a good thing about it too, unlike some of the other mesophytic species. But yeah, I'm from Louisiana, so I know what you're talking about. All right, well, thanks again, Wyatt. It's time to move on to our next talk, and we'll be bringing in Graham Frank next. And Graham is a PhD student at Oregon State University in the College of Forestry, Forest Ecosystem, and Society Department. He's going to talk about the role of timber management in emulating wildfires in the Pacific Northwest and supporting ground beetle populations.
Yeah, thanks, Kevin. So as Kevin mentioned, I'll be talking today about a part of my dissertation project that is examining biodiversity in early successional or recently disturbed forests in the Pacific Northwest. So in temperate forest ecosystems where the mature forests tend to have a closed canopy with very little light reaching the forest floor, forests that experience standard placing fire can contribute a lot to landscape heterogeneity for biodiversity. These forests are characterized by abundant deadwood, both in the form of snags and down logs. The abundant light reaching the forest floor allows for the codominance of different plant growth forms. So we see a lot more forbs and grasses, resprouting broadleaf shrubs and trees, all mixed in with conifer regeneration. And that provides important habitat for species that need these open environments. These early seral forests can also be really variable from stand to stand, and that also contributes to biodiversity at a landscape scale. Part of this is temporal due to this rapid successional change. These are forests at 4 and 19 years post-fire. You can see how much difference there is in woody vegetation and the decay of those snags. And part of this variation is spatial. There's a strong influence of biological legacies due to differences in the pre-disturbance forest structure and composition and the actual disturbance effects. And all of that contributes to a lot of diversity across these different stands. In contrast, young forests created by timber harvesting can be compositionally and structurally simplified. So they have very little deadwood due to the physical removal of these logs. Conifers, often a single species, are planted at very even spacing. And vegetation that competes with those conifer seedlings is controlled via the use of herbicides on private industrial land or mechanical means on public federal land. And all of this contributes to more homogeneous composition and structure, both within as well as among different sites. There's still this element of rapid successional change contributing to temporal variability here, but the actual tempo of development is influenced on the front end by that vegetation control, and on the back end by these really fast growing conifers that can quickly create a closed canopy. But at the same time, a lot of the species that utilize these early successional habitats have a pretty broad amplitude for the different habitats that they can utilize. And so we've asked, to what degree do managed forms of early seral support the biodiversity that's traditionally associated with their closest natural counterpart, which in the Pacific Northwest is standard placing wildfire? And recognizing the importance of temporal development in these stands, we've asked this question across a chronosequence of stand ages from 2 up to 20 years after each disturbance. We're also examining the effects of post-fire salvage logging, though due to a bit of a lack of comparable stands in these youngest and oldest age classes, we're only looking at salvage logging in this 6 to 10 year age class. And we're taking a multi-taxa approach to biodiversity here. So we're looking at birds, bees, plants, and carabid ground beetles, which I'll be focusing on today, to get a number of different perspectives on what biodiversity is in this system. And we're thinking about biodiversity both in terms of diversity metrics like species richness that characterize the structure of these communities, as well as the composition of the communities in these different types of early seral. So what species might be overlapping, what species are distinct, and what are their characteristics? So why have we decided to include ground beetles here? Well, for one, in contrast to birds and bees, which are highly mobile and might be responding to conditions in the surrounding area that we're sampling, most ground beetles are pretty restricted in their movements, and so are going to be responding to habitat conditions that are really fine scale. Unlike plants, most of these ground beetles are predators or generalist omnivores, and so are somewhat integrating these lower trophic levels as well in their responses. And we know from studies in other systems that they're sensitive to disturbance and succession, mostly due to sensitivity to forest structure and composition. And even though they're only one family of beetles, they have a wide range of traits that are relevant to disturbance and succession here. For example, you tend to find larger beetles in closed forest environments. Some of these beetles are actually capable of flying and so can quickly colonize disturbed habitats. And as I mentioned, they have a pretty wide range of foraging habits. So we've situated this study down in Southwest Oregon in the Klamath Mountains ecoregion. And we've chosen this landscape for our study for two reasons. One is this patchwork of public and private industrial forest land ownership, which you can see in this checkerboard pattern here. And the second is this series of fires that have occurred in the area between 2002 and 2018 across this landscape. And so we've situated sites in Douglas fir forest types, either on public federal land within these fire footprints in severely burned sites, or in private industrial land in the adjacent matrix at similar stand ages. And at each site, we've placed 12 pitfall traps for 6 weeks in late spring to collect ground beetles. And so far, we've sampled 2/3 of our eventual study sites, collecting over 4,000 beetles representing 35 different species. We're also looking at habitat variables, which I won't get into too much today, but the relevant ones for ground beetles are deadwood transects and vegetation structure and composition. A few things that we're expecting to find here is that management is going to alter the temporal development of early seral forests, and that's going to scale up to ground beetles, and that we'll see different diversity and individual species responses through time after different disturbances. We also expected that management is going to create more homogeneous early seral forest habitat, both within and among different sites, and that this will scale up to the responses that we're seeing in ground beetle communities. And one thing that I haven't really talked about yet is that we expect to see disturbance severity actually differing from the perspective of a ground beetle. So even though these are all stand replacing forest disturbances in that they remove the entire forest canopy, we expect to see that some ground beetles are actually able to survive that clear cutting event residing in the litter layer, whereas through fire coming through and consuming that litter layer, that's actually going to create much higher mortality, probably create more open niche space in that post-fire environment. And so we expect to see really pronounced differences in that initial post-disturbance species composition. Now, I'm going to show some results first on species richness, but first I want to talk about the scale at which these results are being measured. So we can think about biodiversity, of course, within a single stand, like how many species are in this one area. But there's also variation among stands of similar ages. And so the results I'll show for diversity within an age class integrate both that local within stand diversity as well as that variation among stands. And what we're seeing so far is that stand replacing fire here is in orange. These intensively managed plantations are in blue. And we see much higher species richness after fire right off the bat than in clear-cut environments, but it declines quickly through time. And in contrast, the species richness of ground beetles in these plantations is much more stable. And though I won't show the data here, just for brevity, these patterns are really similar to what we're seeing for site level richness, which really suggests that the amount of variation from site to site after these different disturbances is probably quite similar. And to me, this raises the question, on a landscape where we have all of these different age classes of early seral forests occurring together, what are the implications for scaling up to that landscape in terms of the diversity supported by one disturbance or another? And what I'm getting at here is that if we have our most diverse crabid communities in these recently burned sites, and we're just losing species as diversity declines through time, then the diversity on the landscape that's supported by this fire-generated early seral is really just what we're seeing in those youngest burn sites. In contrast, if as diversity declines, we're actually gaining some other species through time, then our overall landscape scale diversity might be much higher. And so here I've overlaid estimates for that diversity integrated across all stand ages for each disturbance. And you can see that for stand replacing fire, basically all of the diversity that we see supported by fire-generated early seral is encompassed by those youngest age classes, which suggests that we're really just losing species through time after fire. Somewhat similarly, we see that a single age class of post clear-cut communities, this older age class, is supporting all the diversity we see across the landscape for that disturbance, but that's occurring later in succession here. And both of these together suggest that there might be pretty limited differences in terms of actually gaining species in any one of these age classes compared to the more diverse age classes. But this is kind of an indirect way to get at how communities are changing through time. And so to examine that more directly, I fit a joint species distribution model for our 13 most common species. And you can see these species names on the left side here. Don't need to worry about what those actually are. The way to interpret this figure is that these dark orange colors indicate strong support for a given parameter in this model for that species, and dark blue indicates strong negative support for a parameter in this model. And I'll point out a few themes here so that you don't have to actually interpret this figure yourself. The first here is that we find some forest-associated species are persisting temporarily in clear-cut sites. So Pterosticus herculaneus here is one good example of that. You can see it's really quite abundant right off the bat in these clear-cut sites, but declines through time. And we find it very little in contrast in the post-burn sites, especially right away after fire. See a similar trend here for Promocognathus crassus, where we almost find it in none of our burn sites, except for a couple of sites that were salvage logged, but it's actually quite common in our clear-cut sites. So these seem to be species that were able to persist through that initial disturbance, but not through fire. Another trend that we're seeing here is that some of our most recently burned sites have a number of species that are capable of flight and are actually, as a result, very good colonizers of recently disturbed environments. One example here is Amanita conflata, which again, you see very common in some of our post-burn sites, but declining through time. And we find very little in the sites that weren't burned at all. It suggests perhaps that due to a lack of competition in these burn sites, these species are able to kind of thrive there, maybe because we're not finding those forest holdovers quite as much. And the last group of species, which accounts for a lot of them actually, are these disturbance generalists. And these are species, I'll just show one example here, that do seem to occur most commonly in the recently disturbed sites, but they don't seem to have a preference for one disturbance over another. It's really just these open environments that they seem to be thriving in. So to tie us back to some of these initial hypotheses, that first, disturbance severity differs from a crabbed perspective. That's really supported by this evidence that we have some forest holdovers initially in clear cuts, and that we find a lot more of these colonizing pioneer species in our burned areas. The second was that management is going to alter the temporal dynamics of these early seral forests. And we do find some evidence for that with our highest diversity supported by burns being right off the bat in these young burn sites, but our highest diversity following clear cut happening later in early successional development. However, this may be mostly because of that difference in disturbance severity, creating kind of a broader temporal variation in burn sites, as opposed to differences in forest structure. And lastly, we expected that management would homogenize early seral forest habitat. And we did find more species supported by fire-generated early seral at the landscape scale, but we did not necessarily find support for the idea that these fire-generated early seral environments are more variable in their species composition from stand to stand. So a couple of next steps here. I'm hoping to integrate trait analysis into this approach so we can actually get an idea of a little bit more of a mechanistic understanding for why ground beetle communities might be differing between disturbances and through early seral development, looking in particular at their foraging niches, their body shape, so like how well are they able to push through some of this dense vegetation, and their dispersal ability. We're also planning a third field season coming up this year where we'll boost the sample size by 50%. So hopefully getting a little bit more power for some of these analyses, being able to look at more individual species in depth. And with that, I'd like to thank in particular the field crews who have helped me collect this data. You can see from this photo that it is not easy work. And also Jim Labonte for helping me with crab and taxonomy, because I wouldn't even know where to start with identifying these beetles without him. And happily would take any questions or suggestions at this time.
All right, thanks a lot, Graham. Great presentation. We don't have any questions yet in our queue. I was just kind of curious about, so when these populations or communities of beetles come in after the fire, are there any surviving the fire somehow, or do you think they're all new, just translocations from other locations in the post-fire environment?
Yeah, that's a good question. So we don't have direct evidence for that because we're really just sampling at individual time points after disturbance. But I think we can kind of infer based on some of these species life history traits. For example, these species that are flight capable that we seem to be finding only in the burned environments, it seems likely that those are colonizing after fire. But even though these severe fires are consuming the entire litter layer in most of these forests, I do suspect that there are some species that are, or at least some individuals that are able to survive that event. We have some species that will burrow beneath the soil. And I think, we know that soil is a really good insulator. And so you probably don't have to get that deep to survive that fire event, particularly if it's moving through there pretty fast. But I think that a lot of them are going to be moving in from adjacent unburned forest, very likely.
Within some of the unburned forests have fewer beetle species. So I guess kind of in a similar related question, the beetle species that tend to only show up in the early successional habitats, kind of, where might they be coming from if they're fairly localized populations, like you said at the beginning of the talk? Does that make sense?
Yeah. So if they're not very mobile, how are they colonizing these burn stands?
Yeah, especially if you tend to find them only in the early successional stands. Like ecological theory would suggest that the ones that show up first are the ones that are the best at colonizing new locations or dispersing, but I don't know if that's necessarily the case with these beetle species.
Yeah, I think that overall is probably what we're finding. A lot of our young burn stands are between 2 and 5 years old, so there is some time there. We're not coming in immediately after the fire and sampling beetle communities, so they, even if they're walking there, they have some time to get there, I guess. But yeah, I think some studies have shown pretty slow recolonization of disturbed habitats. And so I think that there's definitely a lot more analysis work to be done here in terms of what habitat variables are actually really important for some of these beetles. But I'm kind of expecting that because of the rapid regrowth of vegetation that we get post-fire, whereas, after in these intensively managed clear cuts, you get herbicide use that pushes that back. I'm expecting that there are more kind of microsites that are available for some of these forest-dependent species once they come into the burn stands to actually be able to persist there. I guess as opposed to some of these clear cuts that are really large, right? In Oregon, the limit is 120 acres for a clear cut, and most of these private companies are going up right to that limit. And so these are really large patches, and a lot of our high severity forest patches, and this is kind of a mixed severity fire regime, they tend to be at least pretty close for the most part to a lower severity burned area or an unburned area.
So beetles don't have to go as far to recolonize the areas. In other words.
Yeah, so I think there's possibility for more source populations there.
Is there variation among the beetle species in terms of their endemism versus being much more widespread species throughout the West?
Ooh, that's a good question that I don't have an answer to.
It'd be interesting to see if there's any relationship between that and the dependence or preference on recent fire habitats.
That would, yeah.
Because usually more generalist species tend to occupy a more general range of disturbances.
Yeah, that's a good point. Yeah.
Okay, well, thanks again, Graham. We'll need to move on to our next speaker now.
Okay, thank you.
All right. So next up, we're going to have Thea Whitman. She's associate professor at the University of Wisconsin in Madison, in the Department of Soil Science. And she's going to be sharing some groundbreaking research on the effects of fire on the understudied world of soil bacteria, especially taxa that seem to benefit from and maybe even depend on fire. So welcome, Thea.
Thanks. I'm really delighted to be here. This is my first time at this meeting, and I've really been learning a lot from all the sessions so far. Before I get started, I just wanted to start with a few acknowledgements. The work I'll be talking about today, a lot of it comes out of my lab, actually including work from Jamie Woolet, who started us off in this session, as well as collaborators with the Canadian Forest Service and the University of Alberta, and also the US Forest Service with Kara Yedinak. So just to quickly orient you to the region I'll be talking about, for the work coming out of our own lab, it's largely set in the North American boreal forest, specifically up in northern Canada. So this is a really large ecosystem. It's characterized by wildfires. Here in green, you can see the extent of the boreal forest in North America. And in gray, you can see where wildfires have occurred in the last 30 to 40 years. So pretty widely burned. And the fire regime of the Northwestern Canadian boreal forest is generally characterized by large crown fires. Usually it's a few large fires that are really responsible for the majority of the area that's burned. And typically these large fires are ignited by lightning. So as the community here would be well aware, the field of fire ecology is well established. People have been thinking about fire ecology for many decades now. And much of the progress in fire ecology has included plants and then also animals, as we've heard here today. But when it comes to microbes, they're often a little more overlooked. That's to say, of course, when we think about fungi, our knowledge of pyrophilus or fire responsive fungi does go back, over a century. This is a paper from 1909 noting these mushrooms that will pop up post-fire. Particularly when it comes to bacteria, it's also relatively well understudied. And when we think about, for example, plant traits that are relevant for doing well post-fire, there's so much work in this area, right? So we know about traits that allow organisms to quickly recolonize post-fire, things like resprouting or having serotinous cones. Also maybe surviving the fire, thinking about traits like having thick bark or self-pruning, and then other broad characteristics that allow plants to do well post-fire. And we're beginning to think about the extent to which these traits might also translate to the microbial world. So do we also see fast growers, heat survivors, and organisms that do well in that post-fire environment? But this framework is really just beginning to be developed and tested and established. Before I go too far down that road, I thought I would just take a brief moment to maybe try to convince the audience here that you might be interested in microbes. So one of the obvious ones, even if you really aren't interested in bacteria and fungi, if you're interested in plants, you probably would care about microbes. So, many fire-affected environments, the plants often rely on direct symbiotic relationships with microbes such as ectomycorrhizal fungi. So this is a paper by Sidney Glassman and others, where they looked at the ectomycorrhizal fungal spore bank post-fire, and in this case observed that it remained largely intact. If we're thinking about bacteria, some of recent work led by Jamie actually, we looked at short versus long interval reburns up in northern Canada, and found that this taxon had decreased abundance in the short interval reburns versus the long ones. And this is interesting because Caballeronia sordidicola has actually been identified by other researchers in BC as being able to exist as an endophytic nitrogen fixer in a few different conifer species. And in greenhouse trials has been demonstrated to fix meaningful amounts of nitrogen, contributing up to 50% of these little seedlings nitrogen in these sort of optimal conditions. So in addition to plant interactions, microbes, of course, drive really critical global elemental cycles, whether we're thinking about carbon cycling and the role that bacteria and fungi play in that, or if we're thinking about nitrogen cycling. So just as an example, Monica Turner's work out in Yellowstone has observed this sort of unaccounted for source of nitrogen in the lodgepole pine that's been regenerating. And current thinking is that this source may be due to nitrogen fixation by microbes, but that's an explanation that's remaining to be explored. And then also on the sort of direct human impact side, we might think about compounds such as mercury. So mercury can be volatilized during fires, but then microbes can transform the mercury and particulates into methylmercury, which is a potent neurotoxin that could then enter the food chain. In addition to elemental cycling, global biogeochemical models are increasingly including microbes. So this is a model by Ben Solman and others, known as the corpse model, that directly represents microbes. And some of the work we're doing is trying to integrate fire into this model. And then finally, given that this is a biodiversity system, soils and microbes contribute a really large portion of global biodiversity, depending on how we think about that. One of my favorite facts is always that in just a handful of soil, there are more microbes than there are people on Earth. And so that's sort of an extraordinary diversity that exists there. Okay, so what do we know about microbes and their role in fire? So one of the first things, maybe kind of a no-brainer, fire changes soil microbial communities. Of course, fire directly through heat kills microbes in soils, but we also think about the indirect effects of fire on microbes, such as changes to soil moisture, soil nutrients, soil carbon, and soil temperature. And furthermore, these effects are mediated both by vegetation, both pre and post-fire. This meta-analysis by Yamina Pressler and others looked at just changes in microbial biomass or organismal biomass post-fire. And in general, with fires, you see a decrease in biomass, whether you're looking at bacteria and fungi specifically, or other soil organisms. And then in terms of not just biomass, but community composition, in this figure, I'm illustrating some work from our group up in Northern Canada, where you can see here, this is representing bacterial communities. And then the triangles here are the communities from unburned sites, whereas the circles are communities from burned sites. And you can see a clear differentiation here. Also plotted here are open wetlands, which of course have distinct microbial communities. And then the points are colored by pH, and you can see a nice gradation of pH there, which is a factor that is regularly found to have important impacts on structuring microbial communities. And this was data just 1 year post-fire. Okay, so fire changes soil microbial communities, and it also can take decades for them to return to their post-fire, to their pre-fire states. So this is work by Nicholas Dove and others looking at fungal responses to wildfire using a chronosequence approach, where they found that returns to fungal richness and mycorrhizal colonization could take decades to get back to pre-fire levels. Building on our work up in Northern Canada, here we're plotting how communities changed between 1 and 5 years post-fire. So the light colored circles represent burned plots 1 year post-fire, and each point is joined to a dark colored circle, which is the same plot 5 years later. And again, we have our unburned sites here in triangles. And what you can see is that in general, 5 years post-fire, the communities are shifting to better resemble the pre-fire or the unburned communities, but they're still not there yet. So likely full recovery to the pre-fire conditions would take much longer than this 5 year time span. Additionally, again, probably unsurprisingly to the audience here, we know that fire regime and severity also moderate the effects of fire on soil microbial communities. In sort of the broadest sense, this is again from the Pressler meta-analysis, where they looked at the effects of fire here just on fungal biomass across different biomes. And then also, you can see here within the forest, prescribed fires versus wildfires had different effects on the changes to fungal biomass in that burn. Jumping back to our work up in Northern Canada, we see that the dissimilarity from unburned sites increases with increasing soil burn severity. So in the highest severity sites, you see the largest changes in bacterial and also fungal, but not pictured here, community composition. Beyond the entire community, we're also starting to identify specific taxa that emerge post-fire. So starting to think about, like what are the jack pines of the microbial world? And when it comes to fungi, some of these have been well known for centuries, for thinking about fungi like morels or some of the cup fungi that are really characteristic post-fire. But we're also starting to add to this list organisms not visible to the human eye, such as bacteria from the genera Blastococcus, Arthrobacter, and others. And Arthrobacter is an interesting example of a specific bacterial taxon that's a noted fire responder. So this figure here is illustrating the enrichment of taxa 1 year post-fire on the x-axis versus their enrichment 5 years post-fire. So organisms up in this quadrant were enriched 1 year post-fire and remained enriched in the burn plots compared to unburned 5 years later. This circled one here being Arthrobacter. And so it's interesting to see this, but what's especially interesting to me is that this organism has been found to be a fire responder in ecosystems that are really different from Canada's boreal forests. So these are studies from Spain in oak forests, where we see that Arthrobacter, in some cases has gone from being essentially not detectable in unburned sites to making up more than 20% of the community. And also similarly in this study, being most enriched in the highest severity plots. So it's kind of a really strong fire responder. And so as we're identifying these organisms, it begs the question, what are the specific traits that allow these bacteria to emerge and become more abundant post-fire? And so to look at this, I'll draw on some recent work in our group where we were linking lab experiments with field data to identify pyrophilus traits in bacteria. So first, we have field data from these 2014 wildfires up in Northern Canada, which we sampled 1 and 5 years post-fire. I've already shown a couple of plots from this study. And then we returned there to collect intact cores, which we brought back to the lab for experiments. And so in this study, we took intact cores and subjected them to heat fluxes designed to represent that of a crown fire in the region in a cone calorimeter at the Forest Products Lab here in Madison. And essentially, we had cores collected across a wide range of sites. And then we subjected them to both wet and dry burns to try to sort of explore a range of temperatures being experienced during the fire. And then first of all, we looked at which taxa survived the fire by looking at ribosomal RNA that was enriched just 24 hours after the burn. And then we also looked at, empirically speaking, which organisms are capable of growing quickly by incubating them for 5 weeks and seeing which organisms became more abundant. So we use these two approaches to, again, empirically identify which organisms are potential fire survivors and which organisms are potential fast growers. And then we looked back into our field data for the same organisms to see how abundant they are 1 and 5 years post-fire and across burn severity. So starting with fire survivors, this figure here is showing data for 1 year post-fire and 5 years post-fire. And we're looking at the relative abundance of all the organisms that were identified as fire survivors across this soil burn severity index. And what you can see here is that, both 1 and 5 years post-fire, you see a significant increase in bacteria that are fire survivors in both of these years. With these effects remaining particularly notable in the organic horizons 5 years post-fire. Jumping over to our next trait, fast growth, a similar plot here. And again, you can see a really strong response here in 1 year post-fire where these fast growers are making up, in some cases over 40% of the microbial reads in this study. But interestingly, this trait seems to be most important just immediately post-fire, where 5 years post-fire, we don't see these organisms as being enriched anymore. So coming back to this figure, I wanted to talk briefly about another specific organism. So this is a Pseudonocardia, which is enriched both 1 and 5 years post-fire. So it's a strong burn responder. And going back to our lab-based trait incubations, we see that it's significantly enriched in our sort of fire survival treatment and in our fast growth treatment. So it potentially could be surviving the fire. It could also potentially be growing quickly post-fire. But in addition to this, we've isolated a Pseudonocardia from our lab, within our lab. You can see here a bunch of different organisms growing on this black Petri dish. It's black because the only carbon source here is pyrogenic organic matter. And we've used incubations with isotopically labeled pyrogenic organic matter to demonstrate conclusively that this isolate is able to degrade pyrogenic organic matter, plotted here against an uninoculated control. And so thinking about this sort of hypothetical microbial trait triangle, Pseudonocardia is an interesting example of an organism that might do well at surviving the fire, might be able to grow quickly post-fire, and also may have sort of an affinity for that post-fire environment due to its ability to degrade fire-generated compounds. So that said, there's still a lot that remains to be explored in this area. So one of the big things we'd be interested in understanding is, of course, the functional implications of changes in microbial community composition, especially thinking about things like vegetation reestablishment post-fire or biogeochemical cycling that's mediated by microbes. It'll also be interesting to continue to explore global patterns in these fire-responsive taxa, and then integrating these fire microbe soil vegetation interactions into Earth system models. And then sort of overarching all of this, ultimately predicting how changing fire regimes might affect all of these above factors. So with that, I'd be happy to take any questions.
Thanks so much, Thea. Wonderful presentation. We have one in the queue. It says, do you know of any studies of microbial changes in fire suppressed sites where the fire has been— I
can't hear you. I don't, because I muted my computer, but now I can, sorry.
Okay, sorry. This is from Rob Klein. He says, do you know of any studies of microbial changes in fire suppressed sites where fire has been reintroduced for restoration purposes? I'm wondering if those kinds of microbial changes could limit the restoration process.
Yeah, that's a really interesting question. I do know in the chronosequence study by Dove and others that I mentioned there, those sites were the fires that occurred were sort of outside of what the historical fire regime would have been because of historical fire suppression preceding the fires. And so that, I guess, would be an example of a study where one might be able to think about how is that affecting recovery. And they did see in that study along that chronosequence, recovery was relatively slow, at least in terms of fungal communities. It's an interesting question. We have to think about things like, where are the microbes coming post, like recolonizing post-fire. And of course, sometimes we think about, the fire completely sterilizing the soil. And of course, at the surface, we can see hundreds of degrees Celsius and complete destruction of their habitat if we're thinking about the organic horizon. But just a few centimeters below the surface, we could have microbes readily surviving it. And so thinking about that recolonization from below would certainly be possible. But yeah, then it's interesting to think about, if the system hasn't experienced a fire for such a long time, would you start losing these fire responsive microbes? And I don't have a super conclusive answer to that question.
Yeah, it's really hard to know where they're coming from because it doesn't take many microbes to recolonize a population necessarily, and it could be way below detection, I suppose. Did you have a question, Wyatt?
Yes, sir. Do you think an increase of fire-loving fungi microbes can increase the site index, and that those species can maybe be used as indicators for a better site index for lodgepole pine?
Oh, that's interesting. Okay, so I'm revealing my fire ignorance here. I don't know what site index is.
So a site index is commonly used for a lot of species. For example, in loblolly pine, a site index, if it has a high site index, that means it can grow loblolly pine trees really well, because the soil is best suited for it, or there's a lot of other factors.
Okay, nice. And So you're wondering if fungi would serve as indicators or interact with that?
Yes, and increase, make it better for it.
Yeah.
So
yeah, that's a great question. Sorry. So, I mean, certainly others have found that, like, for example, ectomycorrhizal fungi are often depleted post-fire.
yes.
But then I guess there's always a bit of a chicken and an egg question there. You know, are we really just seeing that they're not so abundant because their hosts are not present? And, you know, a few years down the line, will we see that abundance increase again? Versus, is the absence or destruction of those fungi in the fire actually fundamentally limiting the recolonization of those species? I think that's an open question that remains to be like really conclusively experimentally demonstrated. But I think it's a really interesting thing to think about.
Thank you.
The fact that it seems like most of the communities are working their way back to pre-disturbance conditions is at least encouraging, it seems, because it could go to a completely novel state after such severe disturbance, it seems like.
Totally.
Well, thanks again, Thea. It's time to move on to our next presentation. And next, we're going to have Johanna Freeman. And she's an associate research scientist at Florida Fish and Wildlife Conservation Service in the Fish and Wildlife Research Institute. She's going to be sharing research on the relationship between plant community structure and insect pollinator use in sandhill pine savannas in the southeastern U.S. and how we might better use fire regimes for pollinators.
Hi, thanks, Kevin. All right, I'm going to share my screen.
Okay, got it.
All right, and all right, is that good? Does everyone see my?
Looks great, Johanna.
Okay. Okay, so as Kevin said, I'm Johanna Freeman, and I'm the terrestrial habitat research team leader at the Fish and Wildlife Research Institute, which is a division of the Florida Fish and Wildlife Conservation Commission. That's Florida's state wildlife management agency. And today I'm going to be presenting a portion of the results from a multifaceted research project involving plants, pollinators, and fire in longleaf pine savannas. On my introduction slide here, I've got a photo of a recently burned longleaf pine savanna. It's got an herbaceous understory, a sparse midstory, and an open pine canopy. These are fire-maintained savannas. In the absence of frequent ground layer fires, they succeed into oak-dominated closed canopy forests. These savannas were once widespread in the coastal plain of North America, but the extent is now much reduced and heavily fragmented. The plant community composition varies a lot between longleaf pine savanna fragments. There are a lot of known and suspected reasons for that. We know that vegetation structure, fire history, and soil properties are among the key drivers of plant community composition. We expect that those differences in plant community composition among fragments will affect pollinator species composition as well, due to differences in floral resource provisioning. I'm going to give a little background here to give you the management context of this study. So what I'm showing here are two different expanses of longleaf pine savanna in adjoining regions with preserves cut out of them. We've got natural variations in the plant community between preserves due to all the factors I mentioned in the intro. And then on top of that, we've got fire suppression differences over the years. So let's say preserve 4 doesn't get burned as much. We get a woody dominated structure and we lose some of our original understory community, and we might just not know what would have been there. So now we have these 4 preserves, and this is how our land managers encounter them. They're in isolation from their historical context and even in isolation from other present day preserves. These are burned and managed with general guidelines, but as standalone entities. My research group at FWRI is heavily integrated with the management side of Florida Fish and Wildlife, which conducts prescribed burns and ecological restoration treatments on tens of thousands of acres each year. What these guys always want to know from us, especially with a fire suppressed situation like the one on the right, is, are we there yet? Do we have a good plant community? Do we have the right habitat elements for such and such animal? Those are really difficult questions to answer. And in recent years, I've been going toward a meta-community approach to trying to help answer those questions for each preserve. Looking at this as one big flowering plant community across many fragments. In the study I'm presenting today, we're asking those questions about both plants and pollinators. This was a large study with a bunch of collaborators, and a big part of it was plant pollinator networks, which is not what I'm going to present here. For this talk, I'm focusing on vegetation structure and its relationship to overall flower abundance and pollinator abundance. Those are coarse metrics, but they're really useful for conservation purposes, and they tie in well with fire management. So these are the hypotheses for this component of the study. Flower abundances will vary between savanna sites. Differences in flower abundance will be driven by vegetation, spatial, environmental, and fire history variables. And pollinator abundances will in turn be driven by flower abundances. So we had a bunch of criteria for selecting sites. We had 9 different preserves in North Florida and Central Florida. They're all actively managed with fire for at least 15 years. No history of intensive agriculture or forestry. Old growth indicators in the understory, and the presence of gopher tortoise burrows. This study was focused on longleaf pine sandhills and integrating into clay hills, and the gopher tortoise is an indicator of those subsets of xeric longleaf pine communities. So I'm going to go over the framework for the study and the methods on this slide. So what we're primarily interested in this study is pollinators, their abundance, their diversity, their flower interactions. And to understand pollinator habitat use, we have to understand flower abundance, diversity, and interactions. There are a lot of different things that we think probably drive flower abundance and diversity. Vegetation structure being one, and then I've also got direct effects on pollinators on this slide. We measured that using both LIDAR and ground measurements. And then seasonality, we addressed that by doing monthly sampling throughout the growing season. That was sampling of both pollinators and flower counts. Fire season, we standardized all of our plots to be roughly 1 year since fire. So because we only had 24 plots, we wouldn't have had that much sample size if we had tried to include time since fire as a variable. So we tried to factor it out as much as possible. We know it's something that's very important in these systems. We tried to standardize it about a year since fire. Soil productivity, we measured monthly soil moisture, and then we also took soil samples for clay and pH. Geographic location, we did a spatially hierarchical design, which I'll show on the next slide. Fire regime, we compiled 30-year fire histories for each site with the dates of all prescribed fires. And the surrounding landscape, we collected aerial imagery using the same drone that did the LIDAR in a 500-meter radius around each plot. And then the understory plant community, we sampled in a grid of 5-meter by 5-meter quads that were laid out across our 1 hectare sampling plot. And that plant community included wind-pollinated plants, which are very important in these communities as well, grasses and oaks. Although a lot of the analysis I'm going to show, we focused only on insect-pollinated plants, we did sample the whole community, and we'll be asking questions about that as well. So to go over our methods in a little bit more detail, we had a 1 hectare plot. We had 24 of these. That's 100 meters by 100 meters. And we had a grid of plant quadrats laid out across that, in which we identified every plant, and we gave it a 1, 2, 3, or 4, depending on how many quadrants it occurred in. We had a ground level structure quad in which we measured shrub cover, shrub height, etc. And then we did 30-minute pollinator transects in each quadrant of the hectare, in which we captured every insect that we encountered pollinating a flower and identified the flower, put it in a vial. And we did that across the whole hectare. So for each of those plots, they were organized within preserves, and they were at least 1 kilometer apart. And then our preserves were at least 10 kilometers apart. And this is what the whole study looked like. We had three different, we're calling them regions. They're kind of mini regions, although those differences are significant for plant community turnover in Florida, even though they might not look all that far apart. The LiDAR work was done with the Gator-Eye Unmanned Flying Laboratory. That's Eben Broadbent at UF. And here's just a visual representation of what that data looks like between a plot with heavy canopy cover on the left and really light canopy cover on the right. And then he gave me a fun animation. So you can see what data we're getting within this plot. This one doesn't have super high resolution at the ground layer, but that resolution actually is present. I'll show that on future slides. And that's what the point cloud looks like for an individual tree. So we got really, really detailed structure data from this. So here I've got some of our structure results. And we took the leaf area index strata from different layers of the forest and then used those in ordination along with ground layer herbaceous cover, because the drone collected LiDAR doesn't get below 50 centimeters, but we did herb cover at that level, and we felt that it played well with the LAI variables, so we include that in this ordination. So what I'm showing here is principal components analysis. Two axes described 80% of the structural variation among plots, and then we did k-means clustering on that, and we found four clusters significantly different from one another. And to try to explain what we're seeing here, I am calling these in green over here herb-dominated savannas. Those are at the high end of an herb cover gradient, which is on the letters are a little small, but that's the biplot arrow there is herb cover. And then I'm calling these tree-dominated savannas. These were, I'm still struggling with what to call them, but I'm calling them tree dominated here. They had more mid-story and overstory than the other plots, and less of the shrub layer. These are shrub dominated savannas, and these are woodlands. These were not very well represented in our sample, but we're actually going to be doing more of this work, another round, adding more plots, and I'm going to bring in more of that part of the spectrum. And here's what these look like on the LiDAR. That's an herb-dominated savanna. The maroon is herbaceous plants. Here's a shrub-dominated savanna. The blue is shrubs, a tree-dominated savanna, and then a woodland. Then I looked into what's driving those differences in plot structure. And going into this, I had that whole suite of variables that I showed on the methods slide. And what I did model selection using the ORDI two-step procedure and found that the best fit RDA model for drivers of plot structure retained fall and winter fire, spring and summer fire, and soil moisture. And we had only one significant RDA axis, which was basically a gradient from high to low soil moisture and spring summer fire to fall winter fire. And then I've color coded this plot. It shows that shrubby savanna and woodland plots were skewed toward the high soil moisture and dormant season fire end of the spectrum. Herbaceous savanna plots were skewed toward the low soil moisture and spring and summer fire end of the spectrum. And tree-dominated savanna plots were variable, but they were centered around the middle of the axis. What I'm showing here, we're going to get into the flower abundance part of it. And I'm using the same color coding from these different types of savannas that I identified. And then I've got high, medium, low, and very low flower abundance quantiles. And this is just a visual representation to show you how variable the overall flower abundance was between plots. And then you can see that the herb-dominated savannas were skewed towards the high end of this axis, and shrub and woodland more towards the low. Those differences were not significant, but I have just seen a lot in here that makes me think if we had a larger plot sample size, that maybe we would see significant differences between those types. So going into the next round of sampling, when we add more to this data set, I'm going to continue exploring these different categories. Then getting into our pollinator results, we had about 350 different species of pollinators that we caught on about 120 different species of flowers. And looking at some of our broad aggregate measures, total blooming plant abundance, that was the count of all plants in flower every month that we went out there, was pretty strongly correlated with total pollinator abundance. And it was also pretty strongly correlated with total pollinator species richness. Now, there's way more to that story having to do with networks and species compositions and all of that. But I feel that these aggregate measures are useful for conservation, for wrapping our heads around whether we have good pollinator habitat on these sites. And these correlations do suggest that things that we can do to promote flowering in the understory are also going to promote both pollinator abundance and species richness. Then looking into the drivers of those, of the flower abundance and pollinator plant abundance, I'll start with insect pollinated plant abundance. That was from the quad survey. This is regardless of whether the plant was in flower or not. It was just its abundance within the plot. What we found was the best fit model retained shrub layer LAI and fire season diversity. Both of those were positive effects. And the reason for the shrub layer positive effect is probably partly related to the fact that a lot of shrubs produce flowers that are strongly attractive to pollinators. Saw palmettos, blueberries, roots, several other things. And then flower abundance, that's the monthly flower counts. We found that tree layer LAI had a negative effect and insect pollinated plant abundance had a positive effect. So a few conclusions about that. The role of shrubs in pollinator habitat provisioning seems to be complex, and it may be mediated by fire season diversity. We're still trying to wrap our heads around what those results mean. That was a Simpson's index that we used on the fire season, but we looked at it all kinds of different ways, and that effect persisted. So it's possible that shrubs, depending on the species or how fire suppressed the site is, they could have a negative effect on habitat provisioning. But maybe if you've got a site where something about the fire regime is promoting coexistence of shrubs with other pollinator plants, maybe that's a positive effect of having shrubs in there. Another conclusion, insect pollinated plants may persist in the understory despite higher overstory LAI, but the flower production might be suppressed relative to more open sites. So that was that effect where we found that the abundance of pollinator plants, regardless of whether they were in flower, was a significant predictor of flower abundance, but it wasn't the only one. Tree LAI also had a negative effect. And we think that those two things might be both playing a role in flower production. I'm concluding that management targets aimed at promoting herb cover are likely aligned with good pollinator outcomes, because we saw herb cover showing up in a lot of those results having to do with flower abundance. And then additional, there's a lot of other work going on with this, things that are going on right now and that we're going to do. Path analysis, a lot of the stuff that I've shown, there are both direct and indirect relationships going on. So with the flower abundance and pollinators and all that, we're going to be using path analysis to look at the effects of environmental variables on plants and insects, both directly and indirectly. Network analysis. One next step that we're taking is to do multi-level models looking at 20 key plant species that we've pulled out, and then using our suite of environmental variables to look at the drivers of those species. And going forward, I think that's going to be a good framework for delving deeper into those species and their specific fire ecology to develop some management guidelines for promoting plant species that we know are important. We're going to be adding 18 more plots to this in the coming year. And then we're also working on pyrodiversity treatments on the plots that I just showed, hoping to make that a long-term experiment to try to get a better handle on fire seasonality and its role in this whole system. And that's all I've got.
Okay, thanks so much, Joanna. Beautiful graphics and photographs. What a treat to watch. I think we're out of time, I'm afraid, for questions. So we'll have to follow up with Joanna through our various chats and communications and messaging, the Whova, et cetera. But I want to thank everybody for joining us for the biodiversity series. Is excellent. And thanks again to all the speakers. Hope you have a good rest of the conference.
So much. Thank you to all of our speakers for sharing your expertise and experience with us today. This session will be available in Whova, our conference app, within 2 weeks after the conference ends. Up next, we have a 20-minute break followed by 5 general discussion sessions, 1 special session, and 2 fire circle discussion sessions. To choose the one you'd like to join, use the agenda tab to the left of your screen, click sessions, and click the main session title to join. To join the session offering Spanish and Portuguese interpretation, be sure to select the session titled Fire Behavior. Thanks again to our speakers and enjoy the rest of your day.